Like a nice, natural, awkward silence. That means it's time to get started. My name is Shobha Subramanian. I'm in the Office of Graduate and Postdoctoral Studies here at Michigan Medical School. I'm the Director of Curriculum and Education Initiatives. And I want to first introduce a few people here who are instrumental in making this event happen before I forget to thank them. Maggie Evans, who is the Career Professional Development Student Program Manager. She is exceptional. Everything that you're seeing here is because Maggie worked tirelessly. Catherine Garber, who was here, she will be handling our video and photography. She's our marketing and communications coordinator. Erin Reifler, who is the Postdoc Affairs coordinator. Don't see anybody else from our team. Kevin Calhoun, who also was instrumental in setting a number of things up. So thank you to our team members. And Mary O'Riordan, who is our Associate Dean for Graduate and Postdoctoral Studies, who has the vision and leadership to let us do phenomenal things like Postdoc180. So thank you to everybody, and I want to give everybody a round of applause. So in chorus, I want everybody to say, Happy National Postdoc Appreciation Week. Let's do this together. Happy National Postdoc Appreciation Week. So who are postdocs? So for those of you who are family members here, you may be wondering, who are postdocs and what do they do? And I got this clip from the kind of ever-famous PhD comics, and you can read it. I won't read it for you. But postdocs are kind of trying to find independence while contributing to science, and they are very important to our mission of education and research here at any top university, including the University of Michigan. And this is kind of the more formal definition of postdoc. And on the right, you have how the NIH and NSF defines postdoc. A postdoc is an individual who has received a doctoral or equivalent degree and is engaged in a temporary and defined period of mentored advanced training to enhance the professional skills and research independence needed to pursue his or her chosen career path. And that's exactly what our postdocs here at the University of Michigan also do. They are kind of an intern trying to find their way forward, but they are very advanced in training and are important contributors to our science here. So at the University of Michigan Medical School, we have 600 postdoctoral fellows. Across the University of Michigan, we have 1,400 postdoctoral fellows. As you can imagine, there are 1,400 PhD degree holders on this campus right now doing science and contributing to our knowledge. Like I said, postdocs not only do science, they also write for funding and proposals. They publish their papers in prestigious journals. They present their work in national, international, and regional conferences. And they're instrumental in mentoring our undergrads and graduate students in the lab. So they have a lot of things that they handle day to day. And at the same time, they're trying to figure out where their careers are taking them. That kind of brings me to my role and my team here at the Office of Graduate and Postdoctoral Studies. So I lead the career and professional development team, and Maggie Evans, who I introduced to you in the beginning, she and I are the two-member team who make a lot of these things happen. And for those of you who have not guessed yet, this is the map of Ann Arbor. That looks like a cell. And the way we approach professional development in my team is we start with career awareness, and the idea is that you end in a productive job search. Okay? Along the way, you are assessing your skills, you're building skills and knowledge. I'll come back to communication in a moment. You learn by engaging in workshops and seminars. You're building your portfolio, your CV, your resume, etc. And you may also engage in more experiential opportunities like internships or externships or shadowing opportunities. And like I said, the goal is to have a productive job search and finding a job of your dreams at the end. I said I'll come back to communication in a minute. So we deliberately put communication as a thing in itself, not because I believe or somebody in my team believes that communication is important. There's a number of reports, including one that we conducted here in our office, where we surveyed employers who hire PhD degree holders, our alumni, and asked them, what is the one skill that is instrumental when you hire an advanced degree holder? And communication came time and again as the important skill, regardless of what career you're going to end up in. So we take communication very seriously, and therefore we want to make opportunities like this available to our students and postdocs. Postdoc180 is the competition we are all here for. This competition was inspired heavily by the 3-minute thesis competition that many of you may have heard of or even participated in. The origins of that competition is at the University of Queensland in Australia. And the idea was to challenge PhD degree seekers to present their thesis work in 3 minutes or under to a lay, educated, broad audience. Postdocs clearly don't do a thesis, but they do advanced research. They already have their PhDs. So this competition is kind of a mirror of that with some tweaking here and there. And the goal is that our postdocs will kind of hone their communication skills, build more confidence. They will be able to showcase their research to a wide community, especially our local community, and engage with them. And I'll talk about our judges in a moment who represent many facets of our community, and win prizes. Did I mention prizes yet? So the first prize is $1,000. Second prize is $500. Third prize is $250. And the judges will vote on that, and I'll talk about that in a moment. And you, the audience, pay close attention because you get to vote. And you get to pick a People's Choice Award. And they get $250 also. Okay. So I want to take a moment now to introduce our 3 phenomenal judges. And may I ask the judge to stand up while I introduce and talk about you. Our first judge is Dr. Mary Ann Misek. Dr. Misek is Dr. Misek is Associate Dean for Graduate and Professional Studies at the College of Engineering. She is also a professor of biomedical engineering. She has had many leadership roles here at the university, including serving on the executive board of Rackham Graduate School. And among her many accomplishments is being elected to the College of Fellows of the American Institute for Medical and Biological Engineering. Her lab works on biomedical photonics with a translational research program that seeks to impact patient care by creating non- and minimally invasive optical diagnostic technologies. So thank you, Dr. Misek. Our next judge doesn't need much introduction. His voice is much too familiar to many of us who listen to NPR on our way to work in the morning. It's Doug Tribou, the host of National Public Radio, Michigan Radio's Morning Edition. Doug has been with Michigan Radio since 2016. Before that, he was a Night Wallace Journalism Fellow here at the University of Michigan. Doug has been in the NPR space for a number of years. Before coming to Michigan, he was in Boston at WBUR, where he had his own show called Only a Game, that was NPR's sports reporting show. His reporting has appeared in All Things Considered, Marketplace, and Weekend Edition. And he also has had many appearances on Here and Now. He also worked in the commercial radio space for a little bit before that. So I know Doug is very busy. He wakes up very early in the morning to be on Morning Edition. So thank you, Doug. Our last, but not the least, certainly, is Dr. Jay Warnhagen. Jay is a current postdoctoral fellow in pathology here at the medical school. Among his many accomplishments is serving on the leadership. He was the president for the Postdoc Association up until a few months ago, and he really championed the cause of advancing our postdoctoral needs, both to leadership here within the University of Michigan and to outside bodies like the National Postdoc Association. But Jay is more famous for the poop emoji. That not only won him the first prize last year in this competition, but also the People's Choice Award. So thank you, Jay, for being here. So I'll quickly go over the rules of the game before we start. So we have 8 amazing postdocs here sitting in the front row. And the task they have is to come up to stage here and talk to you about their science in non-technical terms in 3 minutes or under. What I am going to do is I will introduce them. You'll see a slide with their name, their advisor's name, their department, and their title. And once they come up here, I'll introduce them and we'll start the timer. That's when they start talking. And when it's 3 minutes, they have to stop. There is no Q&A. I know many of us scientists have a hard time believing that, but believe me, there is no Q&A. But I mentioned that there's going to be a People's Choice Award. So take mental notes, or maybe even physical notes, whatever is helpful to you, because you will be voting at the end, and you have to keep in mind all 8 talks as to who you want to vote. Okay? And for sake of complete transparency, this is what the judges have in front of them. They are going to be rating them on 3 main criteria: comprehension, engagement, and communication. So they'll be scoring each of the 8 contestants. And when all 8 are done talking, the judges will leave the room, spend a few minutes outside, deliberate, and come back to us with their top 3. During that time, don't leave the room yet. You want to know who the winners are, but you also want to be able to contribute to the People's Choice Award. So we'll have the poll link and all that good stuff up, so you can have your mobile device or your iPad or laptop to vote. So, ready? Okay, our first speaker is Doug Baumann. Doug is from the Department of Biomedical Engineering, and his advisor is Mohammad Falahi Sichani. And the title of Doug's talk is Reprogramming the Command Center for Cancer Therapies. So when I hit the timer, Doug will start talking.
Okay, so today I've decided to go full nerd and liken Star Trek to cancer and cancer treatment. So in Star Trek, we have Starfleet Command. Starfleet Command provides orders to a bunch of ships throughout the universe to carry out missions. On one of those ships, Command receives those orders, relays the message to the control deck, and the crew members on the control deck, they execute those orders, and it affects some outcome. We can think of a cell in kind of a similar fashion. So when cells encounter growth factors, they are essentially telling the cells to divide. The cells relay that message through a series of protein interactions to eventually hit the nucleus. The nucleus is the control deck of the cell. And these proteins in the nucleus, they carry out and execute those orders and affect an outcome, which in this case is cell division. And when cell division is regulated properly and executed properly, it enables people to live long and prosper. Now, the mission of the starship is compromised when evil Spock arrives. And we know that he's evil because he has a goatee. And evil Spock doesn't listen to Starfleet Command anymore. He's simply pushing his own agenda onto the control deck. The control deck executes those orders, and this leads to death and destruction. In the cell, one protein called BRAF, when it's mutated, becomes the evil Spock of the cell. And you can tell because I've given it Spock's goatee. And Spock, the mutant BRAF, it passes on these signals to the cell to constantly divide. And this ultimately leads to unregulated cell growth. And unregulated, uncontrolled cell growth is, of course, cancer. So the intuitive way to deal with this problem is to get rid of the evil Spock of the cell. So we can treat cancer cells with drugs that particularly target the mutant form of BRAF, and this stops the signaling. And in theory, this should kill all of the cells. What happens in practice, in many cases, is the vast majority of the cells are completely wiped out. But in just a small subset, the cells have become addicted to growth. And it turns out that the nucleus has been programmed to continue to grow even without upstream signals. So my research particularly focuses on understanding the proteins in the nucleus that are contributing to this continued growth even without upstream input. My ultimate goal is to design new therapies that can be used to eradicate cancer. Thank you.
Thanks, Doug. Our next speaker is Shijiao Huang. She is from the Department of Molecular and Integrative Physiology in the lab of Scott Leiser. And the title of her talk is How to Die Young at a Very Old Age.
Aging is the natural process we are facing every day. We are all very familiar with aging. Since we were born, we have decades of disease-free life until when we enter the old age. Usually, more than one age-dependent diseases are happening at the same time in one old person, such as cardiovascular disease, diabetes, stroke, and cancer. And it is very hard to individually cure them all. My research is to prevent all these diseases in their old age by targeting their common cause, aging. By targeting aging, we can achieve longer life. Most importantly, we can achieve longer disease-free life. In other words, the goal here is to die very young at a very old age. However, we cannot study humans directly, as we age in the same scale. That's why we use the roundworm C. elegans to study aging. They have a short lifespan of around 20 days and have very similar aging phenotypes as humans. As you can tell very easily from this scanning EM picture, that the worm on the left is very old, and the worm on the right is very young. Luckily, many longevity pathways have been found conserved from the worms all the way through to humans, such as dietary restriction, meaning eating very little or nothing throughout life. Dietary restriction can make 20-day lifespan worms live to 60 days with healthy aging, 3 times as long as their normal lifespan. which is over 200 years old if it is in humans. However, dietary restriction is very hard to stand for human. Believe me, food is one of the most important happiness in life. At least myself cannot resist food. The good news is, by my study in worms on the genetic pathways under dietary restriction, we are now looking for drugs that can target the genetic pathways and mimic the longevity benefits of dietary restriction, with the hope that in the near future, we will just need to take pills, enjoy our food, and can stay young for much longer years. Thank you.
I'm going to give the judges a moment because I realized I rushed through the last two ones, so I'm going to give like 20 seconds so they can take some notes. Sorry. Do you want to go? Our next speaker is Swetha Jeganathan. Swetha is from the Department of Biomedical Engineering, Ophthalmology, and Visual Sciences in the lab of James Weiland. And the title of her talk is Are Smart Glasses Smart Enough?
In Star Trek: The Next Generation, again, a character called Geordi LaForge, who was blind from birth, was actually able to see using the special visor. But that's just science fiction, you say. Such technology is completely beyond the realms of technology. But is it? As Captain Picard would say, make it so. Today, microelectronic technology is used to provide feature-enriched smart glasses, as those shown here. Smart glasses are getting smarter and smarter. They can enhance images and provide contrast magnification. Clearly, more research is needed. As an ophthalmologist, I often see people who are visually impaired. Unfortunately, I have to give them the bad news that the next option is either a cane or, or this guide dog, which is kind of hidden by this image here, sorry. Anyway, which is a very, very hard task to do. Regardless, my research aims to develop smart glasses as a personalized navigation system. Kind of like your car's navigation system that tells you where to go when you get completely lost. Of course, more research is needed to better understand which design features will best meet the user's daily needs. So far, I've recruited 30 patients from the Kellogg Eye Center. After teaching and training them to use these 3 different smart glasses, I examined their perceptions and preferences for these glasses. And using a recorded interview, I then transcribed the data and analyzed the data for specific themes that emerge. Surprisingly, patients stated that the ease of use of these glasses, especially the usability, was as important as the visual improvement. What does that mean? Anyone who's ever bought a universal remote control for an elderly parent knows that all the technology in the world is useless if they can't figure out how to use the remote control. In other words, advances in product design need to consider both usability and visual improvement. When Geordi LaForge first donned these glasses, such technology seemed to be light years away. Luckily for us, we won't have to wait until the 24th century. And luckily for us as well, at the University of Michigan, we have an excellent computer science and also robotics team. With all of us working together to develop novel technology to help visually impaired people, we can help your neighbor, your friend, or even a family member lead more visually vivid lives. Thank you very much.
Thank you, Swetha. Something happened to the lights, and I'm going to just take a moment if we can figure out what happened. Okay, our next speaker is Ashley Kalinski from the Department of Cell and Developmental Biology. She works in the lab of Roman Giger, and the title of her talk is SARM-1, The Tale of Destruction and Repair.
Destruction, the action or process of damaging something so much it no longer exists or cannot be repaired. This is a powerful and negative word, most of the time. But what if I told you in order to repair something, you actually need to destroy part of it in an exact and organized way? And what if I told you the players involved in destruction are just as important as the players involved in repair? Even more so, what if I told you those players are the same? Neurons, the cells of the nervous system, are responsible for sending signals to and from your brain and are responsible for how we think, feel, and move. These are highly specialized cells, and we have over 100 billion of them. Neurons have a cell body, which is like the processing center of a computer, dendrites, which receive information from short distances, and my favorite part, the axon. The axon is responsible for sending information very long distances, like from your back all the way to your toe. And we can think of the axon as a lifeline between a target, which is like your hand, toe, or muscle, and the neuron cell body. And there's constant communication going to and from the target and the cell body and back again. And this isn't just happening with one axon, it's happening with several axons all the time. And a bundle of these axons is called a nerve. And we have over 200 nerves in our body. The one I study the most is the sciatic nerve, which runs down your leg. Now, I usually tell people I study spinal cord injury. It's like my elevator pitch. But the truth is, in order to find ways to repair the spinal cord after injury, I actually have to study something that repairs itself quite well. So unlike the spinal cord, the sciatic nerve, if traumatically injured, can in fact repair itself and regenerate. And that's because it beautifully destroys one half in order to fix the other half. And the half that needs to be destroyed is the half that's no longer connected to the cell body. And the cell body of the axons of the sciatic nerve live in your lower back, and those axons run down your leg. Now, the protein that we think is responsible for this destruction is called SARM1. If you remove SARM1 from mice, nerves that have been injured can no longer destroy themselves. But what we have found is that it's not just required for destruction. The repair mechanisms are also lost without SARM1. So my work focuses on finding out who SARM is talking to in the half of the nerve that is destroyed, and who SARM is talking to in the half that is ultimately repaired. While those signaling mechanisms might seem to be distinct, we actually think they're working together to orchestrate several responses at once. And if we can find out who SARM is talking to, we might be able to find out what's going wrong after spinal cord injury. So my work has shown that SARM is talking to other cells in the area, like immune cells, that are required for both repair and destruction. And it's highly likely that these signals are blocked or turned off in the spinal cord, thus preventing this coordinated repair and destruction process. So at least in my eyes, I think the future of traumatic injury lies within the delicate balance of SARM1 destruction and SARM1 repair. Thanks.
Give the judges a couple of seconds to catch up. Okay. Our next speaker is Zeribe Nwosu. Zeribe works in the Department of Molecular and Integrative Physiology. He works with two advisors, both of whom I see are sitting here in the front, Kostas Lysiotis and Marina Pascadi-Magliano. And the title of Zeribe's talk is Cancer, Winning the Battle Starts with Knowing the Enemy.
I am studying pancreatic cancer, which is one of the most lethal cancers in the world. For example, in the United States, about 50,000 people are diagnosed of pancreatic cancer every year. But the 5-year survival rate from pancreatic cancer is 9%. Think about that. That means that out of 100 patients who are diagnosed of pancreatic cancer, only 9 can survive beyond 5 years. This is a big problem. But the bigger problem is actually that to date, there is no effective treatment against pancreatic cancer. Why is that so? Well, one of the reasons is that the tumor is not just one cell, but it's a mixture of different kinds of cells. If you like, you can call them enemies. And this is why it is difficult to find a treatment that works for most patients. So how do we address this problem? And how do we solve the problem of pancreatic cancer? We need to be able to identify the specific tumors that will respond to specific treatments. And that's what I'm very interested in, looking at genes, gene level in tumors. And here, for example, you can see that even though all of these are pancreatic tumors, they're fundamentally different. Some of them have low level of my gene of interest. Some of them have high level of my gene of interest. This is what I meant when I said enemies. We need to know which is our enemy and which we respond to what. But we cannot gain that knowledge from patients directly. We need to use cell lines to study this in the lab. In addition, we need to find mouse in which we will induce pancreatic cancer. We call these mouse models. And with those, it is much more easier to translate the knowledge we gain to treating patients than it is with cell lines, which is a little bit of an artificial system. So what we are interested in is understanding what kind of nutrients enable these tumors to grow. And we know specifically that some of them are highly dependent on glucose, sugar. Some of them are highly dependent on amino acids, which they use to accumulate proteins. So the goal is to find a way to starve tumor to death by blocking their nutrient use. But we always have alternatives in research. Why not just go directly to find what treatment will work specifically for those tumors that have low level of my gene of interest, or treatment that will work for those tumors that have high level of my gene of interest? So I'm very confident that we can find solutions to pancreatic cancer or other cancers. But I believe that the first step is to identify the specific tumor type we should treat with specific drugs, rather than treating all tumors as one. Thank you very much.
Okay, moving on. Our next speaker is Awanti Sambarey from the Department of Biomedical Engineering, and she works in the lab of Sriram Chandrasekharan. The title of her talk is The Fight Against Tuberculosis, Smart Treatments with Artificial Intelligence.
What's common between mummies and cocktails? Surprisingly, tuberculosis. Let me tell you how. So tuberculosis, or TB, is one of the world's oldest infectious diseases. In fact, it is so old, it's even been found in mummies as early as 600 BC. And here we are, centuries later, with TB now the world's deadliest infectious disease, killing over 1 million and infecting 10 million people every year. Despite its long history, we only identified the root cause of TB in 1882. These very pretty bacteria, Mycobacterium tuberculosis. We now know that they spread through cough droplets and mainly infect the lung, causing lung cavities. And over time, treatments have been super interesting, from special diets to nutrition to rest to extremely painful lung collapse procedures. Thankfully, post-World War II, we moved on to a more agreeable treatment, a cocktail. Yay! Well, a drug cocktail, where a combination of 4 drugs are given for 6 months. Now, 6 months is a very long time, and these drugs have some adverse effects. So many times, patients end up stopping their treatment prematurely. And this has allowed the bacteria to evolve and acquire genetic changes that allows them to resist these drugs to which they were once sensitive. So clearly, we need to look at developing newer, more effective cocktails to overcome this problem. But doing so experimentally is a challenge. See, if you were to design an optimal 4 drug combinations from just 50 drugs, you would still need to do at least 200,000 drug interaction experiments. That's simply impractical. So this is where computer science, artificial intelligence, and our research comes into play. Using a type of artificial intelligence called machine learning, we have built a model called Indigo. Now, to Indigo, we feed in information about different drugs, dosage, properties, interactions, as well as Mycobacterium genetics, mutations, growth patterns. Indigo then runs thousands of simulations to identify common patterns, and it then predicts new treatment combinations for TB. It also predicts if these drugs in combination will work together in synergy, or if they will oppose each other's actions, what possible effects they may have, and what pathways they affect. Our wonderful collaborators here then experimentally validate some of our best predictions. So with this integrated smart approach, we hope to develop new, more effective cocktails for TB, which will help us combat this dreadful disease that has otherwise outlived all of us. Thank you.
So for those who are keeping track, we have 2 more speakers to go. So our second last speaker is Mary Underwood from the Department of Pediatrics. Mary works in the lab of Carl Diesch, and the title of her talk is The Cloth Thickens.
So I'm not from around here, I'm from the UK, and in order to come here to Ann Arbor to do some science, I had to take a long-haul flight. And I'm sure many of you have taken long-haul flights before, and like me, you probably get up regularly to walk around for reasons other than asking the flight attendant for another drink. So we get up and walk around because long periods of inactivity, such as on a long-haul flight or after an operation, can increase a person's risk of developing deep vein thrombosis, as depicted by the diagram on the top left. So blood clotting is an important, normal, finely balanced process. We want our blood to clot enough so that we don't bleed after vessel injury, but we don't want it to clot too much that our vessels become clogged. And in deep vein thrombosis, this balance is tipped, so there's excess blood clotting. And these blood clots can actually break off from the region in which they form in the leg and travel to other parts of the body, such as the lung, where they can lead to blockage and to the fatal disease pulmonary embolism. So we know some of the factors that are important in the development of this disease. So we have environmental factors, such as inactivity, but we also know that genetic factors are important. So certain mutations or abnormalities in certain blood proteins important in the normal blood clotting process can increase a person's risk of developing disease. However, there are a number of unidentified genetic factors. And so in order to identify those, our lab went back and compared the DNA of patients with this disease to healthy controls. And along the way, we identified mutations in a gene not previously associated with this disease, with the charming name STAB2. So the mutations that are actually present in these patients reduce the amount of functional STAB2 protein. So the question now is, what is STAB2 and how can its deficiency lead to this disease? So this is what I've come here to Ann Arbor to try and work out. So STAB2 is a protein present on the surface of endothelial cells. So these are the cells that line the blood vessels. So this protein is in direct contact with the blood. And this protein is also known as a scavenger receptor. So scavenger receptors can target old proteins or old cells for degradation. So we can think of it as akin to a garbage collector. So what happens if we don't have enough STAB2? Probably the same thing that happens when the garbage men go on strike. So we have a buildup of unwanted material. In this case, this could be old proteins in the blood. And as the clotting process is a finely balanced process, any abnormalities in the amount or quality of proteins can potentially tip the balance, leading to thrombosis. So part of the work that I'm doing is trying to understand what STAB2 interacts with, aka what this junk material is. So we are at the beginning of unraveling the mystery of how a deficiency in STAB2 can lead to this disease. But by identifying the factors that STAB2 interacts with, we may potentially find new explanations as to why the clot thickens in these patients. Thank you.
Okay. Our very last speaker is Prahatha Venkatraman. Prahatha is in the Department of Pharmacology, and she works in the lab of Manoj Putanvide. And the title of her talk is, Should We Be Worried About Mild Injuries to the Head?
Hi. By show of hands, how many of you remember the classic Nintendo game Super Mario Brothers? Awesome. For those of you that don't, this game had a lot of bricks as part of the decor. As a child, I had discovered that if you repeatedly hit these bricks, you get coins. So I made Mario headbutt a lot of bricks. I didn't get many coins. If you're an 8-bit character like Mario here, you will walk away from the seemingly mild head injury and proceed in your quest. But what if Mario is a human? Did all that repetitive brick breaking permanently damage his brain? Now, we may not headbutt bricks on a daily basis, but we are exposed to scenarios that may cause mild injuries to our brain. Yet we don't make a big deal out of it because we don't think it's causing us a permanent damage. What if someone told us that a mild injury, if repeated several times, makes us more prone to diseases like Alzheimer's 20 years down the line? Now, that becomes something to be very concerned about. A lot of the research in the field of traumatic brain injury is aimed at understanding how severe injury can affect brain function, like in the case of sports injuries. However, I'm interested in studying how repeated mild injury can affect brain function. The easiest way to test this would be to take a brain in a dish, expose it to repeated mild injury, and studied over time. But having a brain in a dish is easier in a cartoon than in reality. So in my research, I grow cells in a 3D environment to mimic conditions inside the brain. I then create mild injury to my cells using a very sophisticated device we developed. Again, not the hammer shown here. I then use advanced microscopy techniques to look for changes in how the cells function in response to this mild injury. By varying the magnitude and the frequency of the injury, I can better understand this complex relationship. In future, we can even include patient-derived cells to look for personalized answers. So I hope that my research can create an injury threshold scale. What does that mean? Going back to the example of Mario, I hope I can predict to him that headbutting bricks more than, say, 100 times in a period of 6 months will permanently alter his brain. So the next time you bend down to pick up a fallen Sharpie in your lab and you bang your head against your workbench, or when you go home and your very excited dog just rams into your head, remember, I'm researching to find out if you'll be all right. Until then, don't headbutt bricks, okay? Thank you.
Okay. So this is the part when the judges are going to leave. So once they have left, I'll post the link where you can start voting. I know it's going to be a tough choice if it was what I'm feeling right now. All the talks were so spectacular, so it's going to be tough. All right. So if you have a laptop or a phone or an iPad or any kind of internet-connected device, you can go to pollev.com/pibs, or there are some mechanisms to text. And we just put the images of the 8 presenters, so you can jog your memory quickly before you vote and cast your vote. Should be in any minute now. The judges are having a hard time. I got a text, so bear, bear with us for another minute or two. And I can't imagine why. Should I say the clock thickens? And if I forget to mention later, there is a nice reception outside. So please, when this is done, go out and mingle with your colleagues and peers. If you have questions for the postdocs who presented, they will be outside. You can ask them questions about their science. Yeah. Hey, there they are. We're just fighting. I can believe that with you, Jay. Suppose it went to sleep. Or I'll chat with her friends. So that's the people's choice. Right. So I want to, as Maggie is writing, we're just looking at the people's choice on the computer. She's writing the name down. I want to thank all the participants. Hey, are we ready? Okay. So the third prize goes to Doug Baumann. And he'll get his $250 in. I'll tell you how you're going to get it after the event. There's no check in that. The second prize goes to Prahatha Venkatraman. And the first prize of $1,000 goes to Awanti Sambarey. And who did the people pick? It's one of these three. I'm killing you right now, I'm sorry. It is Prahatha Venkatraman. So thank you everybody for attending. This was a great event. And go science, go blue, go postdocs. And thank you to all the postdocs. So there is a reception. Please enjoy the reception and mingle with your colleagues and postdocs. Yeah. Thank you so much.